No problem. Okay, thank you so much for giving me the opportunity to speak today. It's been an amazing day so far. There's been lots of great presentations, so no pressure or anything. But today I'm going to talk to you about sustainable labs and a bit of a kind of a starting point for a guide for us to start moving towards going green in the lab. Thank you for the introduction. Yeah, I'm a sustainability architect. And so this is just a bit of an overview of what I'm going to talk about today, a brief introduction, talk a bit about sustainability in labs. I'm going to touch upon the plastics pledge and single-use plastics, a little bit about my work as a sustainability architect, a few kind of hints and tips and thoughts regarding reduce, reuse and recycle, and the laboratory efficiency assessment framework for anyone who's not heard of that, known as LEAF as well, and then sort of a wrap up and a conclusion. So a little bit about me. I'm a second year PhD student, so I'm based in the School of Chemical and Process Engineering and the School of Physics and Astronomy, and I'm looking into sustainable materials consumption, materials development, specific composites, natural fibre-based materials. I've got a background in fashion and chemical engineering, and my interest in sustainability has brought me to working on a really great project as an architect this year. And so, as we know, obviously the kind of SDG goals are forming a pivotal part of our conferences, and climate action is quite an important one to kind of focus on at the moment, and we're tackling climate change and its impact. And so reducing carbon emissions and looking at the way we work more efficiently is a major part in reaching this goal, and it does involve a very collective effort across kind of the way that we operate and the resources, materials we use, and also thinking about that full life cycle of the things we do use and how we dispose of them afterwards. Sustainability in Labs, it's an initiative by the University of Leeds, and it's working to improve the efficiency of how we work in our lab spaces, and help us to make the most sustainable choices that we can within the limitations of what we need to do for research with regard to materials and energy use. So there's goal 13 that we're obviously focused on, but we're also we're also linking into the SDG goals of 7, 9 and 12, which is looking at affordable and clean energy, infrastructure, industry and innovation, and responsible consumption and production. So we know that laboratory work is essential for innovation and discovery, and the University of Leeds is at the forefront of research and creating a lot of high impact research each year, but it's very resource intensive. So for example, there's a lot of energy and water use, so it can take a lot of energy to run specific equipment, maintain certain temperatures as well. 4 times as much water can be consumed in a lab when compared to that of an office, I found out recently from some literature work. And there's also materials consumption and waste generation, so they're kind of going to go into that in a bit more detail. But you know, that laboratory activity accumulates a lot of waste. We use a lot of materials that we have to throw away. The nature of lab work, there's a lot of contamination issues and risk. You're working with hazardous chemicals, working with biological samples. We need to make sure that for reproducibility and quality of that research, that we haven't got contamination. Also, in terms of disposing of that waste as well, we have to dispose of it through certain measures, and that usually involves incineration at high temperatures. It's very difficult at the moment for us to kind of put that through any other route that would allow it to be recycled. There is some work in that area, but at the moment that is kind of a major challenge. So it is a top priority for you, the university, to help move forward in tackling climate change and achieve these goals. But as I said earlier, it's a collective effort, and we really don't have all the answers. I'm not going to tell you the perfect solution today, but hopefully I can give you some insight into the things that I've been learning as part of this project, and a few hints and tips that I'd obviously welcome some feedback on. So there's some ways we can work more efficiently, and also there's this opportunity to discover better ways of working. So one thing I've sort of realised while I've been doing this work is that there are certain things we're not sure of. We don't know if we could wash certain items and get the reproducibility that we need for our research, but maybe that is a research project in itself that a sustainability architect that's assigned to a specific school could do, or a master's project student could be supervised by postdocs, for example. So there's a lot of opportunity to actually generate high impact research in order to also find out the best ways that we can work efficiently. So for those who aren't aware, the Plastic Pledge is a collaborative pledge made by the university in 2018 to remove single-use plastics across all operations by 2023. So it's 2022 now. Two of the biggest contributors to the carbon footprint of plastic actually comes from, number one, the incineration at the high temperatures, but also from the production of new plastic items, which in some cases has been shown to contribute to around 90% of that total carbon footprint in actually just producing a new plastic item. An estimated 80% of total plastic waste in the UK is generated from laboratory activity alone. That's quite a lot. And plastic manufacturing, as I said, it's very energy intensive and it also requires raw materials derived from non-renewable fossil fuels. The single-use plastics, I'm sure most people are sort of aware of what they are, but it's an item made of plastic intended to be used only once or for a short period of time before we dispose of it. So obviously laboratories use a lot of single-use plastics, and these items are essential to the contribution of our scientific research. I can't just walk into all the labs at university and say stop using all your plastic. It's kind of got to be this sort of process where we understand what people are doing, and so we need to think about, you know, is there ways that we could reuse? Could we reduce the use of it? And is there some sort of research we need to do to work out if we can recycle certain items and what we need to do in order to make that a reality? And are there feasible alternatives that could replace the items? It's kind of finding out about that and looking at sort of reuse, replace, recycle, reduce kind of holistically. So historically, glass was a primary choice for lab-based items. In kind of last decade, a lot of glass items have been replaced by single-use plastics. They are cheaper to produce, they're durable and strong, they can be moulded into different useful shapes for research. Plastics have also got a lot of favourable attributes such as corrosion resistance. They can be temperature stable, they can withstand higher temperatures that some plastics can, for example. So as I mentioned, I'm a sustainability architect, and so part of my project was to actually just explore at the beginning why different single-use plastic items are used across the university. We have sort of 50+ labs in this university, all working on lots of different work, and it's important for us to understand what items are being used and why they are being used, because there'll be different reasons across different labs working on different projects and different samples, for example. So I looked at doing an initial lab audit. Now I was going to follow this link, but I don't want to mess up I don't want to mess all the technology up, so I'm just going to talk through it. But I did an initial lab audit, first of all, in my own department, and just looked at kind of plastics that we're using in the School of Physics, just across a few labs, got an idea of the kind of key plastics that are being used, did some sort of research into other items that were maybe not being used in my lab, and I started to develop a questionnaire that the aim was for it to be user friendly for lab users to fill in and asking questions about what plastics they do use, why they're using them, have they looked at alternatives, is there any barriers they think to actually changing using that item. And so I kind of put this together in this sort of user-friendly form that actually ended up being huge. I think there was about 50 questions by the end of it. I did some iterations, I got some great volunteers to give me some feedback, because, you know, there are areas of science that I'm not aware of. I'm very much in a sort of engineer background and very physics, so I don't do a lot of work in biological things, so there was a lot for me to learn and get some feedback on that. So just initially, the really nice thing is that there is a willingness to make change. People are trying. There are a lot of labs across the university where people have really actually tried and said, well, yeah, we tried this, but for reasons X, Y, Z, it's not worked. You know, but there are small changes being implemented across labs to sort of help. Cost and time, major barriers, particularly in research. A lot of people are up against kind of deadlines. PhD students, postdocs have got time. There's time that's not on the side all the time, and costs as well. So, you know, single-use plastics are cheap, convenient. There is that risk with buying certain glass items. They might break, and then that might be an extra cost that they don't have. As I mentioned, sterility is very important in labs. So reuse is a concern for some samples. Are we sure exactly how clean that item is when we've cleaned it, and what is the best way to clean it? Some plastics can't be autoclaved. I think might be wrong on this one, so don't quote me. But I think polypropylene can be autoclaved it can be autoclaved at high temperatures, but I think PVC struggles. There's also PTFE that can obviously withstand high temperatures, but there are some other plastic types that can't. So it's important to sort of know which one you're working with. And it's just difficult to replace some single-use plastics. We don't have glass pipette tips. You know, they are a pipette tip and they're plastic, and you know, they sometimes can't be replaced. And as I mentioned, glass is not always suitable. So certain cells behave differently on glass surfaces. There are coatings that are added to plastic items to help with things like cell culture. So reproducibility is an issue. Some contamination. It's easier to just throw something away and use something fresh. And then like concerns for breakage, health and safety, that kind of thing. Everyone was encouraging about getting more education and training involved. So it's been really nice to hear about, for example, integrating sustainability into the computing curriculum, and also hearing about from sort of the work in the School of Medicine as well. That kind of thing everybody's really sort of encouraged about. Okay, so reduce, reuse and recycle. There are 3 R's of environmentally responsible behaviour. So exploring them across laboratory work could help us reduce waste associated with single-use plastics. I kind of explore that a little bit, and as I said, I don't have all the answers, the perfect answers. It's more about things that we're kind of thinking about. So if we reduce the amount of plastic, we can help to massively reduce the impacts of plastic production. Now, obviously, plastics are still being made, but if we can all collectively start to reduce our demand for plastic, there won't be that much demand for more plastic to be produced. And hopefully over time that can help to reduce a kind of global carbon footprint from making so much plastic if the demand's not there anymore. So could we replace an SUP, a single-use plastic item, with an alternative, for example, using glass petri dishes? This has been done across a few labs within the university. There are some flasks that you can get in glass form, certain shapes that would work for you. There are also glass pipettes, depending on whether that would be suitable, if it's something that you could reuse. Paper weighing boats as well. So this has been implemented across the university in some labs, replacing paper weighing boats with plastic ones, FSC certified paper. So it's making a more sustainable choice there. Could we downside our plastic? So that's kind of just a simple thing of thinking about, you know, do you need a big syringe for a small volume? Could you use a smaller pipette tip? Again, it's about sort of that demand for certain larger plastic items could overall start to reduce the need to produce as much. Could we use one large dish to culture small similar cell samples, or could we use smaller dishes when we've got smaller samples? So it's kind of all about these kind of things to sort of think about. There are things to consider. So glass is generally suitable for autoclave, and as I said before, not all plastics can withstand it. There's certain processes, again, as I said, some glass can't withstand extreme temperature changes, so there's difference between soda glass and borosilicate glass. Soda glass is not that great if you've got conditions that do require extreme temperature changes. It can start to affect its accuracy and also damage it. So there's things to think about there. And as I said, we sample behave differently, affecting reproducibility. So there's things to think about that have kind of been sort of challenges for people. Right, reuse. So if we reduce plastic even just once, again, it reduces the amount we throw away and reduce the amount of plastic that you actually need. So, so could we reuse pipette boxes? So a few labs, instead of buying a brand new tip box with pipette tips in, they just refill with a bag of bulk tips. This is happening quite a lot in the university, actually. I've only sort of seen a few kind of comments, sort of people saying, oh, this is something we could implement. So instead of getting a box of sort of 96 tip boxes in a fresh box every time, that box is just being reused. Because most of the plastic involved in pipette tips is actually coming from the box. Could we wash and reuse certain items? So if you're using things like buffers, certain lab work, things like syringes and well plates, if you're working with buffers and things, you might be able to reuse without that affecting your work. How clean do you need your items to be? So do you need to autoclave, or would just disinfecting work? And is it feasible in terms of time and cost? Could you plan that into your lab activity? Could you combine what you need to be autoclaved with other lab users? Things like that. Can we reuse gloves between experiments? So there are some more durable gloves you can buy that would last through multiple use if you know that you actually can just you can clean a gloved hand and use that again. Obviously that won't work if it's a flimsy glove and it will break anyway, but if you can get more durable gloves to kind of allow that to happen. So just a little bit on decontamination. There's kind of 3 types, and the 2 main ones that are I'm going to talk about: sterilisation and disinfection. So sterilisation is the process used to kill all microorganisms, your viruses, your bacterial spores, and that's usually using things like heat, like autoclaves, chemicals, so certain bleachers, and radiation. So that's really kind of your serious, absolute killing everything. Then there's disinfection, so that reduces the number of organisms and contamination to a level where they don't pose a threat to health, but it won't get rid of things like spores. It's interesting to kind of know the difference between these 2. Obviously most people working in a scientific lab will be aware of that, I'm sure. So recycling of lab plastics is complicated. It requires a lot of support, but some steps can help make a difference. Packaging could be recycled. It's not been actually touching any sort of samples that would cause contamination issues. and so they could go into recycling waste streams. Plastic bags containers, you could reuse containers to put things in. You could use bags to recycle and put things in. There's also some companies that offer tip box recycling services. For example, Appleton Woods, if you have tip boxes that you don't need anymore, they will recycle that. So there's things out there that are sort of starting to emerge that will help us, but it is a big process with recycling. Experimental planning is something that a lot of labs have sort of started to implement, and that can help reduce the number of plastics that you're using in the lab, and we can implement that into our training and inductions as well. We can combine orders with other users, that could reduce packaging and unwanted bulk hide. If you don't need 100 Petri dishes, you could sort of share with a larger amount of users. Things like master mixers, so people who work with like PCR work, so serious sort of lab work, could prepare master mixers, and that can allow them to use less pipettes. It might not work for everybody, but some people have sort of done that. Preparing culture media in bulk, that can also be a useful way to, again, reduce the amount of pipettes and things that you're using, and also increase the usage of sort of consumables. If you can, like I mentioned, you know, using bigger dishes so you can fit more things into a larger space, or using smaller dishes to put the same cells into. So glass pipettes could be used in some cases, but you've got to ask, you know, is precise accuracy required? Is reuse possible with that? There are things, you know, there's also a cost thing with that, and that's, you know, something that we've got to really be aware of, that, you know, actually, if reuse is not possible, then you get single-use glass, and then you've got the incineration and the end of life, so you've not really solved the problem. So there's things to think about there. Again, as I said, pipette take back schemes, Appleton Woods and Starlab for some examples, and using tips directly from the bag. And so some lab work, it does allow for cleaning and reuse, and so it's about what support does that lab need from us, from me, to sort of help kind of bring that forward a bit. You could wash a gloved hand to allow the glove to be used for longer. Use thinner gloves, as I mentioned, if you need to regularly change, or use thicker ones for multiple uses. There is some ongoing research into take-back schemes and how feasible a glove contamination idea could be for our campus. There's sort of a lot of ideas out there, but there's limited at the moment in terms of how much it would work, but we're looking into it. ecoSHIELD gloves, an example of the double layer. You get 150 gloves per box, so they've fitted more gloves into a box to just reduce packaging. It's a similar price. Obviously gloves have gone up after in the light of the pandemic, so the price is not too much more, it's quite comparable. They use 60% less ink on the boxes, so to actually remove that dye, there's a lot of energy required in de-inking, so the idea is that it'd be less energy to recycle that. They use recycled cardboard packaging to reduce carbon emissions, and the supply chain uses maritime transport over air transport, which can reduce pollution because it's got a low carbon footprint. Right, so very quickly, because I don't want to run out of time, but the Laboratory Efficiency Assessment Framework is just a little promotion here. It's a Green Lab certification tool designed by the University College London. It's to help us drive improvements in laboratory efficiency. So you can sign up with your lab group and you gain access to a set of criteria that's next to an award. And if you can tick all the boxes of the criteria and write down the reasons how you are matching this, you get an award. And there's 3 levels. There's bronze, silver and gold. And it covers all areas: waste, recycling, equipment management, procurement. So it's a really great opportunity for a lab to sort of see actually what they are doing really well, and maybe there are areas that they could improve. It's a silver award, which is after bronze. The first piece of criteria there is the lab has assessed its use of consumables and implemented realistic measures to reduce waste. And so this is about targeting single-use plastics. So this is quite a big thing kind of across universities. So just to finish off, so my next steps as an architect, I'm still kind of ongoing in this amazing project that's just so much fun, but very challenging to sort of find out the right ways to do things. But I'm looking into compiling a hub for lab users to kind of refer to for tips and advice of how to increase their efficiency and reduce consumption in labs, and give them some ideas about certain items that they use. And I've also started to streamline my questionnaire now to something that's a little bit shorter, and hopefully getting the information we need. So I'm kind of learned a lot from the iterations I've done. If anybody wants to fill this form in, please let me know. And spread the word about Leaf to more labs, and listen to any feedback on changes implemented. This is a long process. We're only at the beginning, and I'm more than happy to get feedback, good or bad, on the things that we're going on about. So thank you so much. Here are a few references. Thank you so much for listening, guys. Any questions, I'm more than welcome. Thank you.
Thanks, Ashley. That's great. It's great to hear about all of that activity. And so I think on the back of that, just interested in what you've found as being sort of most significant in driving behaviour change? Obviously you kind of mentioned the hub, and obviously the networks, I imagine, are kind of key to that as well. But I just wondered what you thought was kind of most significant in sort of encouraging that.
I think the will's there, and I think sometimes when it comes to behavioural change, that's sort of one of the bigger barriers. But people I think a lot of it's habit as well. You get into a certain habit of working in a certain way. You sort of automatically take your gloves off and put another pair on. And I think it's that kind of like, now that it's a big subject and we're talking about it more, if we keep talking about it and we implement these things into inductions and refreshers, we have posters, things like that that could go in the lab to remind people to sort of develop the habits into sort of more efficient habits. So I think that that's sort of one of the things, a key thing that can kind of help, because I definitely think that the will's there and people want to make change. Like, for example, the poster that I've done will hopefully put that in our labs in physics, just as that reminder that this is what we're talking about all the time now. And if you're seeing that and constantly, it will sort of help to implement that into people's minds and be thinking, oh yeah, actually, yeah, I never thought about that.
Thanks, that's great. If anyone's got any more questions for Ashley, if you want to put them in the chat, I think, yeah. Alistair, do you want to put your question in the chat? And yeah, I noticed, Ashley, that you put your email address there. So I guess if there's anybody else that wants to kind of connect with that hub and Absolutely, kind of learn more about it, that they can reach out to you.
Definitely. Oh yeah, I can see the our gloves are £38, but yeah, absolutely. This was the thing I found out really interesting about ecoSHIELD, because at first glance you think, yeah, gloves are at £5 for a box of 100 and ecoSHIELD's 15. You get 150 in a box, but in the light of the pandemic, the glove prices have gone up. So £38.50, that is a wow, yes. Yeah, that is really expensive, and everyone's sort of thinking, yeah, actually, we might as well go for the more sustainable option.
Yeah. And then so interesting topic, and curious we can implement those practices in our faculty labs using timber and concrete.
So is that timber and concrete actual items, or is it research you're doing on timber and concrete? Because I guess it would go towards the sort of items that you're using to do testing or the synthesis of that. Just like sort of knowing that the items that you are using, and again, it's every lab's different, and so if you were happy to fill in my form, I would happily send you that, actually, because it would be really interesting to get the viewpoint from yourself, because then it's kind of this feedback from new ideas and new bits of lab work that we're doing that will actually really help. Okay. These materials have yeah, definitely, yeah. If you would, what I might actually do is just as a plug, I'll just put the form in the chat if anyone wants to fill it in for me. It's quite a lengthy one, but I'd massively appreciate some more feedback from different labs. That would be really great.
Yeah, we can certainly share that questionnaire, definitely. And then Claire's, it's really valuable contribution to the plastic pledge. Yes, obviously Claire's leading the plastic pledge work, making labs more sustainable. So yes, your support of that work is greatly appreciated. I really enjoyed learning about the many ways that labs can reduce single-use plastic consumption and can be more sustainable. I wondered how Leeds University is currently communicating with labs on campus to deliver this helpful information.
Definitely, I know that we do have updates on labs from the Sustainability Service, and Laura has done some communications about the ways sort of some labs have been doing things, and this is kind of something that we're going to continue. I definitely think that eventually we can reach a point where we can have this sort of centralised sort of hub online to have advice, but also things like posters, some visual infographics that are kind of visually appealing to help people kind of remind them. But also, yeah, there's definitely, if you join the sustainability community on Teams, there's sort of events going on where there's kind of updates on what people are doing in the labs, and I definitely recommend that people attend those as well.
Yeah, Emma's posted about the S Labs group, which is obviously one of those key groups, and through the Blueprint programme, where the schools and service areas develop their 5-year action plan around embedding sustainability, we're looking at how we can share knowledge across those individual projects as well. So we're kind of actively responding to that. Alistair's also asked about what about chemical washing with soaps or acids in relation to decontamination. Just referencing that decontamination was discussed in relation to biological microorganisms. So
yeah, I mean, yeah, that's a really great question, Alistair, and I get like the different things that we need to clean, we need to keep clean afterwards, is again a sort of learning process. So I don't know all the answers at the moment in terms of that, but I guess it's what resources and materials you need to get your equipment after you've used those chemicals to a point where you can reuse them, or it could be recycled in an ideal world once we know more about that. And so if it is a case of you need specialised equipment, or if it's something that you need specialised chemicals, and sort of what are the challenges to help you implement those things into your lab, we can, if we find out more about them, then you know, we can sort of look at whether that's something that the other labs need as well, and then we could look at maybe sharing that a little bit, things like that. So I mean, absolutely feel free to sort of talk to me afterwards and tell me more about that, because I'd be really interested to know just like what you need in order to make that a reality for you in your work with your specific chemicals, because yeah, hazardous chemicals is absolutely a major concern across a lot of particular scientific labs.